When you hear the term genetic engineering, what comes to mind? Probably sterile labs, right? Scientists in white coats, super high-tech equipment. But what if I told you that nature's been playing this game for millions of years? Yeah, living right in the soil beneath our feet is this tiny little organism, a master genetic engineer, a natural-born hacker that literally rewrites the code of life in plants.
Okay, so take a look at this. Have you ever been out in a garden or in the woods and seen something like this on a tree? It's called a crown gall, kind of this lumpy, messy, tumor-like growth. It really looks like a disease, like something's gone seriously wrong inside the plant's cells. And honestly, for over a century, that's exactly what we thought it was, just an unlucky case of plant cancer.
But the real story? Oh, it is so much cooler. This isn't just some random mutation or a simple infection gone haywire. No, this is the result of a microscopic heist, a targeted attack where a foreign agent hijacks the plant's own cellular machinery for its own, well, sinister purposes.
All right, so to really get what's going on here, we have to investigate the crime scene. Let's dig a little deeper into this condition known as crown gall disease.
And this isn't some rare thing you'd only find in a remote rainforest. Crown gall disease is super common. It affects a huge range of plants. We're talking over 600 species, including a lot of stuff we eat, like grapevines and apple trees. The dead giveaway is always that tumor, that gall, usually popping up right near the soil line. For ages, the cause was a total mystery, until scientists finally traced it back to a single culprit, a common bacterium just hanging out in the soil.
Okay, so it's time to meet the mastermind. Let's unmask the organism responsible for this incredibly clever biological crime.
Its name is Agrobacterium tumefaciens, and what makes this little guy so special, so revolutionary really, is its absolutely unique ability to pull off a genetic drive-by. I mean, it doesn't just infect the plant, it actively transfers a chunk of its own DNA directly into the plant's chromosomes. It's basically reprogramming it from the inside out.
Just stop and think about that for a second. This isn't just a bacteria making a plant sick. This is a prokaryote, a simple, single-cell bacterium, permanently changing the DNA of a eukaryote, a complex plant. We're talking about a gene transfer across different kingdoms of life. It's honest-to-goodness natural genetic engineering happening all the time, right under our noses.
So, the big question is, how does a tiny bacterium pull off a hack this sophisticated? Well, every good hacker needs a tool, right? And Agrobacterium has one of the best tools nature has ever cooked up.
And here it is. This is the T plasmid. Now, a plasmid is just a small circular piece of DNA that's separate from the bacterium's main chromosome. The best way to think of it is like a USB drive, packed with specialized software. And this one little plasmid, it contains all the genes the bacterium needs to find a plant cell, break in, and upload a whole new set of instructions.
Okay, so if the T plasmid is the USB drive, then the tDNA, that's the malicious code, the virus file, if you will. This little segment of the plasmid is the payload. It's the exact piece of DNA that the bacterium is going to cut out and physically shove into the plant cell's nucleus.
But wait, how does it actually get that tDNA from its own plasmid into the plant? That's where the virulence region comes in. The genes in this section, we call them rhizogenes, are the hacking tools. They literally build the molecular machinery, a whole complex of proteins that works like a tiny syringe, to snip the tDNA out of the plasmid and inject it right through the plant cell wall.
So why go through all this hassle? What's the motive here? It's all about food. The hacked plant is forced to produce these special nutrients called opines. And here's the kicker, only Agrobacterium has the genes to actually eat them. These opine catabolism genes are the bacterium's reward system. It's creating a private, exclusive buffet that no other microbe can even touch. It's a brilliant, self-sustaining racket, really.
So we've got our mastermind, we've got the tool, and we've got the motive. Now let's walk through the heist itself, step by step, and see exactly how Agrobacterium reprograms its host.
Okay, it all kicks off with an injury. When a plant gets a cut or a scrape, it starts releasing chemical signals, stuff like acetosurungone. For Agrobacterium, these chemicals are like a dinner bell. The bacterium senses them and boom, that activates its rhizogenes, the hacking tools. That machinery then makes a fresh copy of the tDNA, injects it into a wounded plant cell, and finally, the tDNA worms its way into the plant's own DNA, becoming a permanent part of its genetic code. Game over. And once that tDNA is locked in place, The plant cell is under new management. It has new orders. The genes on that tDNA command it to do two main things. First, go wild producing growth hormones. This causes that uncontrolled cell division that creates the tumor, which makes a nice, cozy home for the bacteria. And second, it's forced to crank out those opines, turning that tumor into a personal food factory for the entire bacterial colony.
And get this, Agrobacterium tumefaciens isn't the only one in on this scheme. It's got a close cousin, Agrobacterium rhizogenes, that runs a pretty similar operation, but with a different endgame. Instead of a T, or tumor-inducing plasmid, this one uses an R, or root-inducing plasmid. The goal isn't a lumpy tumor, but instead a massive, uncontrolled explosion of roots.
And the difference in what happens is pretty wild. On the left, you see that crown gall tumor we've been talking about. But on the right, that's the work of the rhiplasmid, a super dense, tangled mess of hairy roots. So you have two different plasmids, two different sets of genetic instructions, leading to two totally different results. It's just an amazing example of specialized biological hacking.
So for the longest time, this bacterium was just a pest, a really fascinating one for sure, but still a pest that farmers had to deal with. But then, scientists had this totally revolutionary idea. What if we could hijack the hacker? What if we could turn this incredible natural delivery system to our own advantage?
And that is exactly what they did. All those decades of research into how this little bacterium works, as this quote points out, became the absolute bedrock for modern plant biotechnology. Understanding this disease literally gave us the key to a whole new world of science.
And the process they came up with is just beautiful in its simplicity. Scientists figured out how to disarm the T plasmid. First, they use molecular scissors to snip out the genes that cause the tumor. Then, in their place, they slot in a gene they do want in the plant, maybe for something like drought resistance or better nutrition. After that, they just pop this modified plasmid back into the bacterium and it does what it does best. It goes and infects the plant, delivering the new, useful gene, but this time without causing any disease at all.
So here's the big takeaway. We turned a pathogen into a precision tool. This natural genetic engineer, which was once just the cause of crown gall disease, is now the go-to method for creating a huge variety of genetically modified plants that are absolutely essential to agriculture and research all over the world. We basically turned its weapon into our workhorse.
It really makes you wonder, doesn't it? We stumbled upon this incredible biological machine just by studying what we thought was a simple plant disease. It kind of leaves you with this big, fascinating question. If nature created this perfect system for swapping genes, what other revolutionary secrets and tools are still out there, just hiding in plain sight, waiting for us to find them?